I had a colleague who was in the Humalog clinical trial who was a very brittle diabetic and had had issues. He was a big guy. And I saw him one day in the cafeteria. I said, Mark, man, you know, wink, wink, nudge, nudge. How's it going? You know, because it's in the trial. You know, how's it going? He says, oh, Jeff. He said, you know, my wife and I love Humalog. And I said, oh, I didn't know your wife was a diabetic. He says, oh, she's not. She just worries about me less.
It's clear from Dr. Baker's story that biologic medicines, like the insulin product Humalog, have a tremendous impact beyond just treating the disease. They can improve the lives of not only the patient, but also their families and friends. But you may be wondering what a biologic medicine actually is. Regulatory authorities, like the FDA, have definitions of what a biologic product is. Their definition is usually quite broad. For instance, it could be a protein, vaccine, blood component, cell therapy, a natural hormone, or a plant or animal extract. That's a lot of different types of products to think about. In the context of this course, we'll only focus on protein therapeutics manufactured using recombinant DNA technology. These drugs have advantages that traditional small molecule drugs don't. They can replace missing hormones or enzymes, are engineered to be extremely selective, and have fewer off-target effects. Because of these advantages, biologic medicines can treat diseases that would otherwise be untreatable. Some examples of biologic drugs include insulin, enzyme replacement therapies, and antibodies, just to name a few. Throughout the rest of the course, we'll use terms protein therapeutic and biologic drug interchangeably to describe products manufactured using recombinant DNA technology. But you should still remember that in the eyes of the regulators, therapeutics manufactured in other ways could also be considered biological products. Today, biologic drugs are one of the fastest growing classes of molecules used to treat disease, growing 3 times faster than other therapeutics. These drugs are extremely complex, though. Since they are proteins, they can contain tens of thousands of atoms. This complexity impacts the structural design of the proteins we will use as drugs and our strategies for manufacturing them. The idea of using proteins as drugs to treat disease is not foreign. In fact, our immune system inherently develops customized proteins for recognizing pathogens like viruses or bacteria when we are infected. These proteins are called antibodies, and they circulate through the blood to neutralize foreign pathogens. Their selectivity and specificity is particularly advantageous in the treatment of disease. Developments during the early part of the 20th century solidified the role these proteins could play as drugs. But it wasn't until 1975 that Georges Kuhler and Cesar Milstein produced a specific antibody in vitro using mouse cells. Now the world could pursue specific therapeutics based on antibodies. This innovation excited the scientific and medical community, and many investigators immediately set out to create antibody-based therapeutics. But remember, the antibodies were made from mouse cells, and it turned out our immune systems do not recognize those as our own. The antibodies did not look human enough to fool the immune system. If instead the antibodies are made to look more human, their effectiveness is significantly increased. This is just one example of how biologic medicines must be engineered and manufactured to work with the human immune system so that they're safe and efficacious. There are many more examples that we'll discuss later, as well as the implications this protein engineering has for manufacturing these therapeutics. Now let's learn about the origins of this multi-billion-dollar industry.